him for his today's talk, the title with Changing Ecosystem of Frozen Earth. And thank you.
Thanks, Amit, and thanks for letting everyone know the years that I got my degree, so that everyone knows how old I am. So what I want to do for the next 45 minutes or so is tell you a little bit about some of the research that I've been involved with in Arctic ecosystems over the last couple of years, specifically since the time that I joined University of Alaska Anchorage in 2011. In my title today, I put the word frozen in quotes, and I did that for two reasons. One is that over the last several decades, we've been recognizing that a heck of a lot more goes on in cold weather ecosystems, even during the winter, than we thought previously. So these systems aren't quite as biologically frozen as we thought previously. And then also because a lot of the climate change that we are experiencing in the far north is influencing winter processes, and those winter processes are likely going to have an impact on what goes on during the growing season. So of course, none of this research takes place in isolation. I've had the privilege to work with some really talented colleagues at Utah State University, University of Alaska, Anchorage, and USGS on a lot of this work. This work has been funded primarily by National Science Foundation. When we work in remote Arctic locations, we contract with a organization called Polar Field Services that handles a lot of the access logistics for remote camps. And I will talk about some work that's taken place in Northwest Greenland. And when we're in Northwest Greenland, we're essentially, all scientists in Northwest Greenland are essentially guests of the US Air Force and the governments of Greenland and Denmark. So I don't think anybody in here would be surprised to hear that climate change is a pretty big deal, and temperatures have been changing pretty dramatically over the past 40 years or so. But the changes in temperatures that we observe are not uniform around the globe. Obviously, the temperature changes are greater over continental areas than over oceanic areas, simply because of the moderating effect that water has on climate. But the biggest changes that we see in temperature tend to be at the high latitudes compared to the equatorial zone. And that's because the Earth's climate system is essentially a giant heat engine that moves heat from equatorial regions towards the poles. So we expect that some of this excess energy in the climate system is going to begin accumulating at higher latitudes. In addition to that, there's a really important feedback effect that partially drives high latitude warming, and that is the relationship between energy interception and sea ice cover. Sea ice is a giant white surface that has a high albedo, a high reflectance, so it reflects a lot of energy back out into space. Well, as warming progresses, sea ice extent during the summer is declining, which results in further energy interception because we're basically changing the reflectance of the northern part of the world. So it essentially winds up being a positive feedback effect. So these are two images that show differences in sea ice extent from September 1979 to September 2016. And over that period of time, there was about a 35% reduction in the extent of sea ice. We're also experiencing more and more dramatic weather events in Arctic systems. If you're paying attention to some of the news a couple of months ago, especially the early part of this winter, it was pretty widely publicized that temperatures over much of the Arctic were about 20 or 30 degrees warmer than normal. This is a photo of water release from the Greenland ice sheet. Water release happens every year from the Greenland ice sheet because it accumulates annual snow which melts during the summer. But during summer of 2012, there was a widely publicized event where NASA had documented that greater than 90% of the surface of the Greenland ice sheet had liquid water on it in July. And this was as far north, this was as high in elevation as the summit of the ice sheet, which is 10,000 feet above sea level. Tremendous amount of water was released during that event. So one of the major hypotheses about changing climate is that it's not only changing water, it's not only changing temperature, it's also changing precipitation. And this is derived simply from the fact that a warmer atmosphere holds more water. So this graph shows trends in precipitation over the second half of the 20th century for Arctic regions. If you're not familiar with looking at the world in this kind of way, this is a polar projection. So you're looking down on the top of the world. So there's Greenland, there's Alaska, and there's Scandinavia. Areas that are in blue have an increasing precipitation trend over the period 1950 to 1999. When you get more precipitation, you consequently increase stream flow. These are data from 1950 to about 2010 showing total annual discharge from 3 of the largest rivers in Siberia, the Lena, Ob, and Yenisei. And here you can see that the rivers are discharging some of their largest volumes over the last 10 or 15 years or so. In addition to these kind of physical changes, we're also seeing changes in the biology of the system. Specifically, we're seeing changes in the timing of spring. So this is a worldwide image of NDVI. For those of you that aren't familiar, NDVI is essentially a satellite measure of greenness. So this is a measure of how much vegetation greenness there is on the landscape. And I'll just draw your attention to 2 areas in the higher latitudes of North America during May 2000 and compare that to May 2015. It's a pretty dramatic change. You can see it again, 2000 to 2015. So spring is advancing. Now the satellite data record, of course, only goes back about 40 years or so. If we want to look at longer term changes, we have to look for other sources of data. And there's a really interesting source of data indicating climate change in Alaska. Winters in central Alaska are long, and winters in central Alaska are quite boring. And for 100 years now, Alaskans have taken to gambling on when winter is going to end. These are data on the breakup date of the Nenana River, not very far from Fairbanks, Alaska. And the breakup date of the Nenana River is known to the minute for the last 100 years because real money is on the line. In 2014, someone who guessed correctly won $363,000. So the data from roughly 1917 until today indicate that spring has advanced at a rate of about 6 days per century. In addition to advancing spring, we're also seeing increases in maximum greenness in the Arctic. So these data show not the timing of NDVI, but peak NDVI, essentially the maximum greenness that is achieved on the landscape over 4 different areas of the north slope of Alaska, moving from south to the north, from 1981 to 2001, indicating a positive trend in greenness over that region. Second graph shows another map, another polar projection map of the world. Here Alaska's on the bottom and Scandinavia's on the top. But areas of anywhere there's a red dot on this map indicates areas where observations have been made of increasing shrub cover. So the vegetation not only is getting greener, we're also seeing species changes on the landscape. Now, a lot of the changes that we're observing in the Arctic broadly are fairly predictable. It's a cold system. It's limited by temperature, and what we're seeing is warming temperatures are leading to more vegetation on the landscape. That's not specifically surprising. But I'm interested in the ecology of a lot of these systems. And ecology, as we all know, is a heck of a lot more complex than that. So I'm interested in, essentially, the indirect or downstream processes or downstream consequences of how these changes are influencing ecosystems. So the major question that I'm addressing, the overall question I'm addressing with my work is, what are the indirect or downstream consequences of changes in temperature and precipitation in Arctic systems? So what I'll do for the next little while is take you on kind of a whirlwind tour of 3 different sites in the northern part of North America. We'll start in northwest Greenland at a site called Tule. Then we'll move southwest to the Tule Lake LTER, and we'll finish up with a study in the Yukon-Kuskokwim Delta of western Alaska. In each of these studies, we use some pretty simple experimental apparatus to alter precipitation and temperature. Both methods are purely passive. We use snow fences to increase snow cover during the winter to simulate a winter with more precipitation. And we use passive warming chambers. Those are basically just clear fiberglass cones with the top cut off of them that serve as little warm microclimates, and we use them to warm soils or advance the growing season. And these chambers are about 65 centimeters at their base and about 35 centimeters at the top. Then within each of our studies, we make various measurements, and I'll address those with when I talk about the data sets. So one of the reasons for investigating changes in Arctic systems outside of climate change is the increasing recognition that winter is not necessarily a biologically dormant season. There's still a lot going on. Data from Hubbard Brook in New Hampshire, the most widely studied or most intensively studied watershed anywhere on the continent, has indicated that there's quite a bit of biogeochemical activity going on in soils, even down as low as 10 degrees below 0 Celsius. In Arctic systems, various researchers have demonstrated that Arctic systems that experience deeper snowpacks actually have higher nitrogen and phosphorus levels in the soil. So these are data from a study about 7 or 8 years ago in central, kind of the central part of the Northern Territories in Canada. It shows inorganic nitrogen and inorganic phosphorus in 2 different zones, a control zone and a snow fence zone with deeper snow in the open symbols. And you can see that the deeper snow zone has higher nitrogen and, in most cases, higher phosphorus availability. In some of these cases, the difference is between 5 and 10-fold. So deeper snow can play a really big role. The question that I was asking in northwest Greenland was, can those changes in winter processes that influence winter biogeochemistry, can they have an influence on plant function the following summer? The study species in northwest Greenland was Salix arctica, the Arctic willow. So this is a very small willow found widely throughout the Arctic, part of essentially the charismatic Arctic microflora. And our procedures here were pretty basic. Actually, our procedures here were pretty basic. We went ahead and measured temperature in snow and in low snow and enhanced snow zones, and then we measure photosynthesis over the course of the summer. So here's a data set that shows some of these temperature differences, and the data show air temperature and surface as well as temperature averaged over several different depths in the ambient and plus snow zones. And what I want you to focus on here is essentially the early part of the winter from roughly the 1st of January until about the middle of April. And here you can see the impact of deep snow versus shallow snow. Areas that received relatively little snow, you can see the temperatures very closely track air temperature at both the surface and depth. And you can see that temperatures reach down to about negative 25 degrees below 0 Celsius. Where snow was deep, we see essentially a decoupling of soil temperature and air temperature, and we see temperatures that are considerably warmer, 10 to 15 degrees warmer for much of the coldest portion of the winter. So we wanted to see if those kinds of influences, if those kinds of changes in temperature during the winter influence photosynthesis during the summer. So this is a photo of Lacey Merrill, one of the undergraduates that spent a summer up in northwest Greenland with me, and she helped make photosynthesis measurements. For those of you that aren't familiar with measuring photosynthesis, it's really pretty simple right now. First thing you do is you spend $40,000 on one of these devices. The second thing you do is you put a leaf in it and push a couple of buttons and essentially get a measure of photosynthesis by monitoring the change in CO2 concentration and water vapor content inside the chamber. So we did that over the course of the summer from roughly the end of June until the beginning of August. The snow in this system's coming off at about the middle of June. And we did that on plants that are in the plus snow zone as well as the ambient snow zone. And what you can see in our photosynthesis data set is plants in the high snow zone clearly have a higher photosynthetic rate than plants in the low snow zone, and the difference is most pronounced during the middle of the summer. In addition to photosynthesis, we can also get data on a parameter called stomatal conductance. And stomatal conductance is essentially a measure of how open the stomata are. How rapidly can water be exchanged between the plant and the atmosphere, and how rapidly can carbon dioxide be exchanged between the plant and the atmosphere. So one criticism whenever you do a snow fence type of study is, how can you possibly make inference on some of these other processes when you're dumping all this extra water into the system? Okay? There's a huge amount of water that went into the system in the plus snow area that didn't go into the system in the lower snow area. Well, one way we can make that inference is by looking at stomatal conductance and asking if stomatal conductance differs between our plus snow and our ambient snow zone, because if there's more water, we might simply expect to see greater stomatal conductance. Well, we don't see greater stomatal conductance. When we statistically tested this, there was no difference between the plus snow and the ambient plants. So that indicates that the additional water was probably not having a very big role. A third parameter that we can look at that's calculated from some of the direct measurements of photosynthesis and conductance is the internal CO2 concentration. So this is basically the CO2 concentration that has to exist substomatal in order for that photosynthesis and stomatal conductance to be observed. And here we observe that these plants are pulling carbon dioxide concentration inside the leaves down lower in the plus snow zones. What that indicates is greater photosynthetic capacity at the leaf level. Well, one way you get greater photosynthetic capacity, in fact, the way you get greater photosynthetic capacity is to simply have more proteins, more chloroplasts, and more chlorophyll. And a good proxy for that is the nitrogen content of the leaves. And what we observe is consistently higher nitrogen content in the leaves in that plus snow zone. The last little bit of data on this study that I'll show you is essentially an estimate of nitrogen cycling in this system. In addition to looking at the nitrogen content, we can also look at the nitrogen isotope composition of the nitrogen in those leaves. The nitrogen isotopes in plants are pretty difficult to interpret, and there's a little bit of hand waving involved, but it's a pretty distinct difference between the plus snow and the ambient zones. Without going into a lot of the isotope ecology here, this kind of a difference largely indicates that plants were deriving their nitrogen from different sources, and this change is consistent with more rapid nitrogen cycling in the plus snow, in the soils that the plus snow plants have access to. So some of these really basic changes in winter characteristics are capable of propagating through this system. We see greater snow cover leads to greater soil temperatures in the winter, which likely leads to greater inorganic nitrogen availability, higher leaf nitrogen, and higher photosynthesis. The link that we haven't made in our studies, but that is a common hypothesis in Arctic ecology, is that this is the process that ultimately leads to greater growth, especially of shrubs, and is possibly contributing to woody plant expansion in Arctic landscapes. Okay, so we want to travel about 1,200 miles southwest to Toolik Lake, Alaska, and take a little bit more of a community level approach. At Toolik Lake, one of the labs that I've worked with in Alaska has had a long-term study where they've examined the influence of altered winter precipitation as well as altered summer soil temperatures or altered summer temperatures on plant community composition and essentially ecosystem structure and function in terms of carbon flux. So in this experiment, we have essentially a fully factorial design. In my notation here, a +0 treatment indicates the control, no changes. +T are plots that have received our summer warming cones, so they have a higher summer temperature. +S are snow fence areas, so they have a deeper snowpack during the winter. And +S +T is the combination of the two. So these data just show temperature for the air and for the soil. The air temperature is pretty predictable. The control and the +S are roughly the same because you wouldn't expect snowpack to influence summer temperature. But the +T treatments show about a 1 degree Celsius higher temperature during air temperature during the summer. Not at all surprising. What has been surprising in this system, however, is the impact of these treatments on soil temperature. Again, these are soil temperatures measured during the summer. And what we see is essentially a linear increase amongst our different treatments, from controls having soil temperatures in the 5 degrees C range to the combined treatment of +snow and +temperature having soil temperatures about 2.5 degrees warmer than that. So both treatments have essentially combined to influence soil temperature during the summer. Now, one parameter that just about everybody in the Arctic, everybody doing Arctic ecology is going to measure at some point in their studies is thaw depth or the active layer. So Arctic systems are permafrost systems. During the winter, the entire soil is frozen, but only some of that soil thaws during the summer. That thaw is referred to as the active layer, and that's where the roots of plants are, that's where biogeochemistry is going on, and that's the biologically active portion of the soil. So what we see is that the 3 different treatments, temperature, snow, and temperature and snow, have all had essentially the same influence on thaw depth. And that is, they all increase the depth of thaw and they increase the rate of thaw over the course of the summer. So what altering winter precipitation as well as summer temperature does is basically alter the hydrology of this system. We can see that change in hydrology if we just look at near surface soil moisture. And these are measurements just made with one of our little handheld soil moisture probes. So it's measuring what's going on in the upper 10 or 12 centimeters of the soil. And here you see, again, a pretty dramatic difference between the control plots and our altered plots. Soil moisture in the control plots is approaching 70%, whereas soil moisture in the other plots is down closer to 50% or possibly lower. So we've had a big change in drainage, at least drainage of the surface soils associated with these changes. So one of the main biological measurements that we've been looking at, or biological properties we've been looking at in this study, is plant community structure. And so every summer or every couple of summers when they make measurements, we basically go out into the plots, drop a point frame, and measure repeated cover at 100 points in each of the experimental plots. We can then take those cover data, combine them in a non-parametric factor analysis, such as a non-metric multidimensional scaling. And then we can use something like permutation and MANOVA to look at differences in community composition between the different plots. So on this graph, we're showing, the first axis and the second axis of that factor analysis. The dashed lines, or the dashed ovals, indicate essentially the 95% confidence ellipse for each of the plant communities associated with the different treatments. And here we can see separation along axis 1 of the control and plus temperature plots from the plots that have received increased winter snow. And that's a significant separation. The species that are labeled on the graph are the major species that changed, driving that change in community structure. So let's focus on Areophorum vaginatum. In the control plots, cover is approaching 70%. That essentially declines as we step up the intensity of the treatments down to less than 20% in the combined snow and warming plots. Salix pulchra also goes in the opposite direction. Increases from less than 20% cover up to about 50% cover. Now, if you're not familiar with your Arctic flora, those names might not mean very much to you, but the common name for Areophorum vaginatum is cottongrass. It's a sedge. Okay, this is a system that has standing water in it for much of the summer, and we've shifted to a salix community, and Salix pulchra is a shrubby Arctic willow species where the shrub comes up to about my mid-thigh in the taller shrub areas. So this is a much drier system. So our changes in the physical environment that altered the hydrology are also having an impact on community structure. Now in 2014, one of the other things that we did was look at the impact of that change in structure on a change in function. And specifically, I was interested in changes in ecosystem level carbon exchange. So this is essentially the ecosystem analog of measuring photosynthesis. Instead of measuring an individual leaf, we would go into these plots and drop a 75 centimeter by 75 centimeter by 40 or a meter tall clear acrylic box over the plot and measure the change in carbon dioxide concentration inside that chamber as a measure of ecosystem level photosynthesis. We then do the exact same thing, but we toss a big dark blanket over it so that we can essentially shut photosynthesis down and measure ecosystem level respiration. So that gives us a couple of different parameters. One is respiration in the triangles, the other is net ecosystem exchange, and then the difference between net ecosystem exchange and respiration is essentially gross photosynthesis by the plants in the system. So again, we have our 4 different treatments and our carbon flux values that are, in this graph, values that are negative are respiration, values that are positive are net carbon uptake. And interestingly enough, in the summer of 2014, we only saw relatively modest differences in function with respect to carbon flux. We see a small increase associated with the temperature plots, but that increase is relatively minor, especially when you're looking at something like gross photosynthesis. It's even relatively minor in net ecosystem exchange. Now it's possible that summer of 2014 was a relatively interesting year. It was an exceptionally cold summer and an exceptionally wet summer. There was snow much of the summer during 2014. So it's quite possible that our differences weren't quite as pronounced as they would have been in a warmer system or in a warmer summer. So these changes have propagated through the system as well. Increasing snow and increasing temperature have combined to essentially alter active layer depth. That alteration, that basically adding water to the system, has had the converse or counterintuitive change of actually resulting in lower soil moisture content. That's altered the plant community primarily by increasing shrubs and decreasing sedges. And at least in 2014, that led to some equivocal changes in carbon flux. Okay, so for our last jump, we're going to take a much shorter trip from Toolik Lake to the Yukon-Kuskokwim Delta of western Alaska. This is a vast coastal Arctic wetland. It's a landscape of roughly 50% water and 50% land. It's flooded by the highest spring tides and the highest fall tides. So if you were here at a high tide in September, you would see almost continuous water across this landscape. Here we take a little bit of a different approach. In the YK Delta, we're primarily interested in how changing phenology of the season interacts with grazing by migratory geese, and if changing the phenology of that interaction influences biomass in the system as well as carbon exchange. Our study species for this portion of the talk is the Pacific black brant. Although as a plant and ecosystem guy, I'm not doing very much with the brant. I'm more interested in what's going on with the plants and the ecosystem. So this is a graph showing changes in phenology, showing phenology advancing over the past 35 years or so. The metric that we're using here is again derived from NDVI, so that measure of vegetation greenness. This metric is day of the year at essentially the 50% point of NDVI max. What this data clearly shows is, again, advancing phenology over the last couple of decades. Phenology has been advancing at a rate of about 2.2 days per decade. So over my lifetime, we're seeing spring occurring close to 10 days, on average, 10 days earlier than it did 35 or 40 years ago. So phenology is advancing in this system, and this system you can kind of think of as the Everglades of the far north. It's a vast coastal wetland that is critical summer habitat for migratory birds from all over North America, and a good number that come from Asia as well. These birds are essentially migrating into a system with a changing phenology, with an advancing phenology. What that means is that they may be arriving into a system that is more phenologically mature now and in the future than they did in the past. So kind of our conceptual model for this study is that there will be a developing phenological mismatch over time because we don't necessarily expect the rate of change in green-up and the rate of change in bird arrival time to be the same. In fact, we hypothesize that green-up is likely to advance faster through time than arrival by migratory geese, because we hypothesize that geese have more constraints on them than the grasses do locally. For one, the geese are migrating from their winter ranges, which are much farther south, where climate change isn't nearly as dramatic. So we don't expect that they're going to be getting as different a cue to begin migration. And of course, the distance is not getting shorter from their winter home ranges to their summer home ranges, and they're probably not going to be able to fly any faster. So we expect that in the future, geese will be arriving into a system that is more phenologically mature. From an ecosystem perspective, what we expect then is that these geese will begin grazing on grasses that have been growing for a longer period of time, and therefore have a higher carbon-nitrogen ratio, are potentially less nutritious for the geese, and what comes back out the other end of the goose is then going to have a higher carbon-nitrogen ratio, which is potentially going to influence the biogeochemistry of the system. So in this study, we take a pretty straightforward experimental approach. We use a 2x4 fully factorial experimental design where we alter the timing of the start of the growing season in half of the plots, these advanced season plots, using those same open top chambers. And then we cross that with controlling the time of the year when geese are allowed to graze in experimental exclosures. So we have an early grazing, a typical grazing that roughly corresponds with the time that geese arrive now, and we have a late grazing and a complete removal of geese from the system set of plots. Then we also monitor what goes on right outside of our experiment in a background plot where the geese are allowed to do and the grasses are allowed to do just what they're going to do, regardless of any alteration by us. Then we make various measurements on biomass and carbon exchange. So here's some data that shows our root biomass for 3 different seasons at the end of the growing season in each of our experimental treatments. So all of these graphs are going to be arranged the same. On the x-axis, we'll have the different treatments with the season treatment on the top row and the grazing treatment on the bottom row. So advanced season, normal season, advanced season, normal season, early grazing, typical grazing, late grazing, etc. The really obvious thing about this graph is that delaying grazing in the system delaying grazing in the system results in quite a bit more root growth. And this isn't particularly surprising. If you remove grazing or delay grazing, Basically, the plants don't have to regrow that above ground tissue, so they're capable of putting more resources into below ground growth. But kind of the surprising thing about this graph is that when you compare advanced to early, advanced to early, advanced to early, the season advancement or the phenology treatment is much, much less important. Changing the timing of grazing is a much, much bigger effect than changing the phenology. The same general pattern is observed with shoot mass. Shoot mass is a little bit more difficult to interpret because most of these plots did get grazed at some period of, all of the plots except for the removal plots, the goose removal plots, did get grazed at some period during the summer. So some of those trends don't come out quite as striking, but you can generally see, again, the trend towards increasing above ground biomass with delay in grazing. Here we see a little bit stronger influence of the early versus, or the advanced versus the normal season, especially in the typical and the late plots. But again, the major difference that we observe has to do with timing of grazing, not so much timing of phenology. So of course, I wanted to go out and measure my carbon parameters. We use a much smaller system here than we did at Tulip Lake. We use a small chamber. This is a photo of Martin Holdridge, one of the undergraduates who was able to spend a whole summer out in the Yukon-Kuskokwim Delta. And he carried around this box full of equipment and a chamber and measured ecosystem level photosynthesis, carbon flux forming for the summer. And the carbon flux data is pretty much what you would expect. Here, I'm actually using a slightly different convention. So I'm showing net ecosystem exchange, but here negative values indicate carbon uptake. So negative values are a carbon sink, positive values are a carbon source. And you can see, again, this trend towards increasing carbon uptake in the system with delaying of grazing and removal of grazing from the from those grasses. Not at all surprising, considering that there's simply more biomass, above ground biomass, in these later grazing plots. More photosynthetic tissue is likely going to lead to greater carbon uptake in the system. And again, we see that the delay in grazing is the larger effect. Changing the phenology of the season doesn't have nearly as big an impact on the changing phenology doesn't have as nearly big impact on net ecosystem exchange. And we largely see the same thing for ecosystem respiration. We see slightly higher respiration with the change in grazing. But for respiration, we don't see as big an effect as we do in terms of biomass or net ecosystem exchange. So changes propagating through this system. Delay in grazing is our dominant change. That results in increased root mass and increased shoot mass. Both of those should contribute to respiration, but we see relatively modest changes in respiration. The increased shoot mass leads to greater gross ecosystem production. And when you combine modest changes in respiration or equivocal changes in respiration with an increase in gross photosynthesis, we see an increase in net ecosystem exchange, primarily in response to changing the timing of grazing in the system. So one other thing that's going on in this system that we really couldn't measure in our small-scale experimental plots is how the geese are likely to use a landscape that may have taller grasses and sedges in it in the future. Geese like to use areas that have been previously grazed. These are referred to as grazing lawns, and the geese are basically able to keep the grasses trimmed to about 2 or 3 centimeters tall through continuous grazing in these grazing lawn areas. They then avoid the taller grasses. So at a larger scale, the big impact in this system may not really be in terms of carbon flux, but may actually be in how the geese use it, especially if the geese essentially decide that this is a less favorable area for grazing, given that change in the phenology. So just a couple of points to leave you with. Overall, our studies have kind of indicated that the direct warming effect probably has a relatively small influence on Arctic system ecology. It's more likely that altering the snow, the winter season snow, as well as the hydrology of the system is going to be more important. And the indirect effects, specifically altered interactions between plants and animals in the system, are potentially the most impactful changes that will result. So the next step is that we have a new National Science Foundation grant that just started in January. And I already have one student, Jess Richard, who's going to be working on that study this summer. And I am in the process of getting another student that will be working this summer. We will be returning to Toolik Lake, where we intend to look at the future nutritional landscape for caribou on the North Slope. And this is a collaboration with 4 other universities, so a very big project. So with that, I'll leave you with your requisite sunset. And I didn't leave much time, but I'll be happy to take a couple of questions.
The unexpected small influence of temperature so far, is that just early into this? If you simulated out 50 years from now, I mean, obviously at some point you're going to get enough temperature where whole ecosystems are going to change. And
So the question was that do I think that the modest influence of temperature now is likely to be larger in the future when it gets even warmer? And probably yes. However, some of these interactions are really, really difficult to predict. So there are likely going to be some more direct effects of temperature that we observe, but I think it's going to be really challenging to predict what the true effect is going to be without some additional studies. And keep in mind, you know, a lot of these kinds of temperature changes are relatively modest. So, you know, we could hit the system harder with bigger temperature manipulations and likely get a bigger effect. But then we also have the confounding effect of going immediately from an ambient to a very, very warm system. And there are some challenges with doing that in Arctic systems. We use passive chambers for a reason, and that is they're really easy to deal with in remote systems. If we wanted to go to more impactful warming treatments, we have to use something active, and that is a little bit more challenging in remote locations. Dan.
So would the geese alter their behavior by maybe staying there longer to keep those grazing lawns, to maintain their body size so that when they fly back, they'll have the energy that they need? What, is anybody tracking the animal side?
Yes. So Sandy's question was, are the geese simply likely to stay in the system later into the year, which would essentially increase that grazing over a longer period of time? And is anyone tracking the geese? So to answer the second question, our camp in the Delta is co-located with a bird migration study and bird rearing study that has been there for 30 plus years. So there is tremendous amount of data on the birds. To answer your first question, I'm not entirely sure if anyone has documented how late the geese are staying in the system, because the bird camp is primarily interested in nesting and chick survival, and they leave the camp in the latter part of July. So they don't have data on that, but USGS might have data on that because they also have, both USGS and Fish and Wildlife do quite a bit of work in the area, and I'm not certain.
On the opposite of that, have they documented that they're arriving earlier? For example, geese and stuff are going to be 2 weeks ahead of time.
Yes, yes. So there have been years of much earlier arrival. And right now, if you regressed green update on goose arrival, you get a slope of about 0.4, which would suggest that the geese are matching changes in spring by 4 days advancing nesting time relative to 10 days advancement of the spring. So they're not quite keeping up as of right now. We do know that you can only advance phenology so much before you're going to start getting into light limitation. So what we don't know is the long-term consequences and whether phenology may advance for a while, the geese may not keep up, and then the geese may be able to catch up because advancing phenology is likely to slow. Any other questions? Probably need to go up the room, so.
Jeff,
Yes?
did you think, for the geese grazing project, you showed the data for the 2004, 2015, and 2016?
Yes.
I noticed there's some data, it does have an air factor, consider long-term factor, not just season, also.
So Shilon's question was differences among the season, and I'll interpret her question as being specifically changes in carbon flux. And what we observed in 2015, 14 and 15 was much greater carbon uptake than we observed in 2016. And we've observed that change in carbon flux across 2 different systems that we use. So we're pretty confident that it's a real change. So what we expect, what we're kind of talking about right now, is whether there hasn't been a major change in the system from our experiments that might be driving that difference. Potentially, we have let the system, we basically let the grasses and sedges grow so much in the system that we might be getting a lot of self-shading now that is reducing carbon uptake. I should see if anybody West River has a question here. Okay, all right, I'm already late, so
Thank you, Josh.